of mRNA. And so, if we look at eukaryotic DNA, remember it's linear. So here's my gene that I want to transcribe. So let's just call it gene X. Pick a gene, any gene. Well, every gene, I should say, is going to have a region of DNA. And this region of DNA, we call this non-coding. What do y'all think a non-coding region of DNA means? So if we're coding for something, that is basically a simple way of saying that we're making protein from it. So non-coding DNA, this is not going to be transcribed, and this is not going to be translated. So it's a piece of DNA, that's all it is. It's DNA, and it's attached to the gene. It's just a non-coding, it's a regulatory piece of DNA, actually. And so out of all of your DNA, I think it's only like 5% of it codes for actual proteins. So not all your DNA is just used to make proteins. There's lots of, our DNA is complicated, and this transcription and translation process are complicated. So you need ways to regulate gene expression, or need ways to regulate what genes are being turned on, what proteins are being made. And so the way we do that are through these non-coding regions of DNA. These are where inhibitors can bind. This is where activators can bind. This is where all sorts of stuff can bind in order to influence either transcription or translation. And so in this case, when we're talking about transcribing, this region of non-coding DNA is called a promoter. And each gene has its own promoter. So gene X is going to have promoter X. The nestin gene is going to have the nestin promoter. The tubulin gene is going to have the tubulin promoter. And so the purpose of this promoter, like I said, it's non-coding, so the gene technically starts right here. And what we see, every gene is going to start with TAC. So this is the start of our actual gene, of what's going to be coded for protein. So again, we call this coding DNA, because it will eventually be made into protein. So stuff that's coded for is going to be made in protein, stuff that's non-coding is not. Anyways, this promoter region is always going to be upstream, meaning more towards the beginning of the DNA molecule, more towards the 5' end. And so this promoter is always upstream from the gene. And what it's going to do is it's basically going to serve as kind of a landing pad for the enzyme that's going to be doing all the transcribing. And the enzyme that we're going to be using primarily is called RNA polymerase. So when we were making DNA, we used DNA polymerase. But since we're making RNA in transcription, the enzyme is RNA polymerase. And so this RNA polymerase, technically it's RNA polymerase 2, this RNA polymerase is going to bind to the promoter. And the reason why is because it has to, before it can start moving down the DNA, it has to get itself ready. So just imagine that this is like the start line for a race. And the runners are getting ready, they're like getting in position, like, okay, we're ready, and then you pull the trigger, and then they start moving forward. So same sort of thing here. They're getting ready to start this whole transcription process. But you need a region that this enzyme can bind to, because the way that this enzyme works, it literally physically binds to the DNA and slides down it. So you have to get it bound to the DNA first before you start making your RNA. So that's the point of the promoter. It's not going to be turned into protein. It's just a regulatory piece of DNA in which this RNA polymerase 1 is going to bind, and then once it binds, it's going to transcribe the gene and make it into RNA. Now, if we said that every single promoter is unique, depending on which gene it's associated with, how can the same RNA polymerase 2 bind to every single different promoter? Because the promoter, it's a unique sequence too. Even though it's not coding for proteins, it's still a DNA sequence. It's not like all promoters are the same. They're all individually unique. And so the way that we get around that is through a region of the promoter, and that region of the promoter is called the ta-ta box because it's made up of a few nucleotides that TA. So every single promoter, every single human promoter anyways, has a ta-ta box. That's what it's called. It's just called a ta-ta box. Has this sequence, this TA sequence. Every single promoter in your body has that sequence somewhere in there. And that sequence, that's the sequence that the RNA polymerase binds to. So that makes sense, right? Because if we have this one enzyme that's going to have the capability of binding to every single different promoter, it makes sense that you've got a region that's the same in every single promoter that basically helps that enzyme bind to the DNA. And that region that's the same, that's the ta-ta box. So the ta-ta box helps the polymerase bind to the promoter. It's basically like, hey, come over here. This is where you want to land, right here. Yeah, because when DNA and proteins interact, it's all based on, complementarity. So you can imagine that this RNA polymerase, it's an enzyme, right? So it's a protein, so it's got all these precise molecular stuff. Well, that has to line up perfectly with this ta-ta box, or else this enzyme can't bind to the DNA. If the enzyme can't bind to the DNA, then you can't transcribe the DNA. If you can't transcribe the DNA, then you get no RNA. You get no RNA, then you can't get any protein, right? So it's all dependent on this first step of transcription. Also, in addition to this enzyme coming in, you're also going to have these other little proteins that are going to come in, and they're basically going to help solidify this process, this starting procedure. And all these they're, like I said, they're just little proteins, and they're called transcription factors. And so a transcription factor is just a protein that's usually going to associate either with the enzyme or with the DNA. It's basically going to help, those transcription factors are going to help make sure that RNA polymerase is bound tightly to the DNA. And so this whole complex of proteins that associates on the promoter, we call that a transcription initiation complex. So it's a complex because it's got multiple proteins in it. It's called the transcription initiation complex because at the very beginning, at the initiation of transcription, you have this complex of molecules, of proteins. So that's why we call it the transcription initiation complex. But anyways, before transcription can begin, you have to form this transcription initiation complex, which is formed between the promoter, the RNA polymerase, and then all the transcription factors around it. And there's multiple different types of transcription factors, and each promoter is going to have a different transcription factor. So in general, these transcription factors are just proteins that help this RNA making process along. And so again, here you can see our gene, so it's double-stranded. Again, we have our promoter region, this non-coding region of the DNA. You have your ta-ta box. This is, of course, where the RNA polymerase and the transcription, where the transcription factors will bind, and then where RNA polymerase 2 will bind. And so you end up making this transcription initiation complex with your transcription factors and then your big enzyme. And then once everything gets started, then the polymerase will start moving down. It'll kind of slowly unwind the double helix, similar to DNA synthesis. And then it'll read that template strand, that 3' to 5' strand, and start making a piece of RNA. So this is just kind of showing the setup of what's going to happen during the initiation of transcription. And I should mention that both transcription and translation, they each have 3 stages. The first stage of each is called initiation. The second stage of each is called elongation. And the third stage of each is called termination. So you're starting transcription or you're starting translation. You're then elongating either the RNA or the protein. And then at some point that process has to come to a stop. That's termination. So they have the same titles, but the processes are going to be different. And so you need to know what happens at each stage. So you need to know what happens during initiation of translation, during termination of transcription, and all the other ones too. So we're going to start here. We're going to go through initiation, elongation, and termination of transcription. So the first thing that's going to happen in transcription is initiation. So in the initiation, 3 things are going to happen. All right, first, of course, you have to have your promoter present, which is it's present anyway, so you don't have to worry about that. Yeah?
One DNA strand have like many promoters?
Yeah, one DNA strand will have many promoters. And so in eukaryotes, each gene has its own promoter. So we have 35,000 genes, got about 35,000 promoters. And again, it's always just going to be immediately upstream, just a little bit before the start of the DNA. So here you can see that start point. That's where that TAC molecule is, that first set of nucleotides in your DNA. And so during initiation, this is basically like everything's getting ready. Nothing's starting, but the race is getting ready to start, so all the runners are getting in their place, getting ready to take off and go. That's what initiation is. And in this initiation, what's going to happen is the transcription factors are going to bind to the TATA box. And then your RNA polymerase II and other additional transcription factors, if necessary, are going to come and interact with the DNA and with those transcription factors already present. That's going to form the transcription initiation complex. Seems complicated, but really all that's happening, transcription factors and RNA polymerase are binding to the promoter at the TATA box. That's all you got to say. So during during initiation of transcription, all right, transcription factors and RNA polymerase II are going to bind to the TATA box within the promoter. Y'all can handle that, right? Okay. Is it okay to move on to elongation? That was pretty straightforward, right? You're just getting everything ready to start making your RNA. And so, again, transcription, we have our promoter here, we have our gene that we're making right here. So before we can start transcription, you're going to bring in those transcription factors and that RNA polymerase to the promoter region. You're just getting everything set up on the promoter. And then during elongation, this is where we actually start making our new piece of DNA. So, you know, the polymerase is moving down, and the polymerase is kind of opening up the DNA as it goes along. It sees a T, it puts in an A. It sees an A, it puts in a U. It sees a C, it puts in a G. So forth and so on until you make your long piece of mRNA. That's elongation, is actually moving down the DNA and making RNA from it. And one thing to keep in mind is that this mRNA molecule is going to be made in the 5' to 3' direction. So another way of saying that is that you can only add nucleotides to the 3' end of DNA. You will never build on the 5' end of DNA. That's true for making DNA, for DNA synthesis. So remember, we had the leading and the lagging, and you know, basically you're not going to be able to add any nucleotides, whether it's DNA or RNA, to the 3' end of any molecule. So that's true for DNA replication, that's true for transcription, you know, it's even true for protein synthesis. So anyways, this is why we always start and use the template strand, which is the 3' to 5'. Because then when we make our new RNA, we're making it 5' to 3'. So we just keep adding on more RNA nucleotides to the 3' end, making the mRNA longer and longer. And then finally, during termination, once it reaches the end of the gene, basically everything just kind of dissociates and goes about its business. So the transcription factors leave, RNA polymerase leaves, the new mRNA is released, and you've just finished transcription. So initiation, you're getting everything ready, you're bringing this transcription initiation complex to the promoter. Elongation, the enzyme's actually looking at the DNA and making an RNA molecule. And then termination, it reaches the end of the DNA, of the gene, and then everything just kind of dissociates, it falls apart. And then transcription is over. And so this is what's actually happening during elongation. Again, we have RNA polymerase II. It kind of is responsible for kind of opening up the DNA a little bit. And then again, looking at the template strand. So if it sees a T, it's going to put in an A. If it sees an A, it puts in a U. If it sees an A, it's going to bring in another U. So forth and so on. So it's real simple how it actually makes this piece of RNA. And again, it's always going to be generated in the 5' to 3' direction, which means that you can only add nucleotides to the 3' end of a molecule. You can never add them to the 5' end. Okay, so we're going to run through RNA processing, and then we'll probably wrap it up right there, and then we'll pick up translation on Tuesday. All right, so after we go through transcription, so we go through initiation, we go through elongation, go through termination. At this point, after transcription, we're going to go through this RNA processing. And again, this RNA processing, this is only in eukaryote cells. It's not in prokaryote cells. So we have our transcription. That's going to be the first thing that's going to happen. The second thing that's going to happen is this RNA processing. And this RNA processing has 3 steps as well. It's not initiation, elongation, and termination. But there's 3 modifications that we're going to make to this protein. And mostly, this is to protect this mRNA that we just made. And so keep in mind that within a eukaryotic cell, we're making our RNA in the nucleus. And so this RNA processing, this is also going to happen in the nucleus. And then after this processing, then the RNA will leave the nucleus, meet up with a ribosome, and be made into protein. But transcription and this RNA processing are occurring in the nucleus. And translation occurs on a ribosome outside of the nucleus. But anyways, this RNA processing, the reason why we do it is because we need to protect the RNA. RNA is a very unstable molecule. DNA, I can isolate DNA from cheek cells, put it in a little tube, and set it on that table for 2 years. Come back 2 years later and still use it in experiments. It's just extremely stable. But RNA, if I did the same thing with RNA, it would be degraded within probably like a few minutes. So you have to be very careful with RNA because there's all these enzymes in the cell that can break down your RNA. We actually have enzymes called RNases that break down RNA. It's all over us. It's all over our fingers. So it's difficult to work with RNA in a lab because you're capable of degrading it all the time. Anyways, so we have to protect this RNA once we spent all this time and energy making it. And so we're going to do that in 2 ways. We're going to add a cap to the 5' end. So you got to imagine at this point, this is the pre-mRNA. So this is the unprocessed RNA molecule, termed pre-mRNA. Or pre-RNA, I think, technically. But anyways, so we're going to add a cap to protect the 5' end. Because again, you got to remember, there's all these enzymes and stuff floating around in the cytoplasm. And a lot of those enzymes are called nucleases, which means they cut off nucleotides one at a time. Obviously, if you start chopping up your RNA, it's going to affect your protein. So we need to add caps to the 5' end and to the 3' end, so that way if these enzymes do come in, it's not actually affecting your RNA, it's just affecting these caps and tails that we added. So the first thing we're going to do is we're going to add this 5' cap to our new piece of RNA. The second thing that's going to happen is we're going to add a little tail, same idea. We're just going to add a little protective cap at the 3' end. But instead of being a cap, all we're doing is we're adding a whole bunch of adenines. And so we call this a poly-A tail, meaning there's multiple adenines at the 3' end. And so that way when these enzymes come in contact, all it's doing is cleaving off adenines, which we don't really care about because it's not part of the RNA sequence. So it's just a way to prevent degradation from enzymes that are in the cell. And so the last thing that's going to happen is we're going to cut out some of these non-coding regions of DNA that are present. And so we've talked about, there's our structure, there's our chromosome. So here's our promoter region that's going to be upstream. And then this whole thing, that's going to be the gene right there. So each gene is made up of alternating sequences called introns and exons. So if we have one gene, we might have exon 1 followed by an intron 1, and then exon 2 followed by intron 2, and then exon 3 followed by intron 3, so forth and so on. So depending on how long your piece of DNA is, you're going to have different amounts of introns and exons.